Hi everybody, and thank you for coming to this session. The title of my talk is Fructose Oligosaccharides as a Novel Cryoprotectant for Mammalian Cells. Essentially, I'm looking at new sugar-based molecules and their ability to cryopreserve mammalian cells. The project that I'm presenting today forms part of a wider EU project which I work on, and the data here is accumulation of work carried out in collaboration with Cytiva in the United Kingdom, where I'm based, CONICET in Argentina, and INRA and AgroParisTech in France. I'll start by giving a brief overview to the fundamentals of cryoprotectants. I'm sure most of you are very familiar with these and how they work, so I won't labour the point too much here. I'll then go into how these new fructose oligosaccharide based cryoprotectants, FOS for short, are actually made, which was a key area for optimisation, before going into how we found they were effective as a cryoprotectant and the optimisation process to determine what form of FOS worked best, before moving on to some results we got in mammalian cells. So, starting with an overview of cryoprotectants. As we know, cryoprotectants are required for successful revival of cells on thaw. They have a wide range of interconnected protective measures, far too numerous to mention here, but some of the key ones include acting as a secondary solvent to allow sugars and salt to dissolve into when cooling. This reduces the toxicity of the intracellular channels where cells reside and so allows greater cell survival. An example of this is shown in image A here from Mazur and Rigopoulos. They can also change the viscosity of a solution to reduce the damage of diffusion-driven processes, and they can also lower the freezing point of a solution and the ice fraction formed at any given temperature, as is shown in image B here. The more CPA, the greater the effect they exert, although toxicity issues also become more apparent at higher concentrations. Cryoprotectants are usually placed into two broad groups, intracellular and extracellular. These do exactly what the name suggests. Intracellular CPAs can permeate the cell membrane and so directly influence the intracellular space. These are typically smaller molecules such as DMSO and also glycerol, which has good cell permeability at higher temperatures. They have a transient effect on cell dehydration and also interact with membranes and intracellular organelles, as well as acting as an additional solvent to compensate for water molecules locked away as ice during cooling. On the other side, we have extracellular CPAs. These include glycerol at lower temperatures as well as sugars. They help by dehydrating cells and so reduce the chances of intracellular ice, and can also provide a protective layer around cell membranes by preferential binding and exclusion. Of course, the most common of the cryoprotectants is DMSO, which has been used since its cryoprotective abilities were discovered by Lovelock in the 1950s. DMSO clearly has many advantages in cryopreservation. However, there are also some substantial drawbacks. It has noticeable toxicity, particularly at higher temperatures and for long addition and removal processes. There have also been reports of negative side effects when DMSO is transfused into patients, which is undesirable. So the question remains, what alternatives to DMSO can we come up with? We propose that one potential alternative in mammalian cells is FOS, fructose-based oligosaccharides. So, what exactly is FOS from a chemical perspective? Well, it's a fructose-based mixture which has been used effectively for the cryopreservation of some bacterial strains. Simply, it is a long-chain sugar made up of fructose molecules. We start with a glucose molecule and add a fructose. This is commonly called sucrose, standard sugar. We then add another fructose molecule to form what we call DP3. We then add another fructose molecule to make DP4 and so on. These can be synthesized using known techniques, some are shown on this slide, and the composition of the FOS mixture can be modulated based on the initial concentration of the building block molecules and the time of synthesis. Once the phos mixture is synthesized, it can be further purified to remove some of the small molecules left over from the production process, the basic fructose, glucose and sucrose molecules. This can be done through several methods, such as by using ethanol and activated charcoal. This can effectively help enrich the mixture with longer chains such as DP3. DP3, remember, is a sugar made up of 3 basic glucose and fructose molecules, and DP4, that is made up of 4 basic molecules, and so on, depending on the final desired composition of the phos. So, we've seen how the phos is produced and the mixture composition can be modulated. Now, how does it work in practice as a cryoprotectant? To answer that, let's move on to the biology. We knew how to develop different mixtures of phos, but that's only the first part. Now, we had to test them in some cells. Here, we looked at Jurkat cells. These are a suspension cell line, a form of immortalized T cells, which is relevant to a lot of cell therapy research. This slide shows a brief overview of the experimental methods. The cells were cultured and then cryopreserved in 1 ml cryovials at 1 degree a minute down to -8 degrees, and then transferred to long-term storage at -140 degrees. They were subsequently thawed, and the cells counted, proliferation ability measured, and the metabolic activity determined out to 3 days post-thaw. Here are some of the results we got, comparing 10% weight by volume of different phos mixtures to our gold standard of 5% DMSO in culture medium, which has been optimized for this cell line. The first thing to note is that the phos can act as a cryoprotectant in the absence of any other cryoprotectants. This was quite a surprise to us. Our initial expectations were that the phos may be effective as part of a wider CPA cocktail, and we were pleasantly surprised when it turned out to offer quite substantial cryopreservation capabilities on its own. The different mixtures here are indicated from phos 1, which gave the worst cellular outcome, to phos 8, which gave the best outcome. As you can see, there was a very wide range of results between the different phos compositions, some offering no cryoprotective ability at all, others becoming comparable with the current DMSO gold standards. We carried out similar tests in CHO cells, those are Chinese hamster ovarian cells, HepG2s, a liver cell line, CACO cells, which are a colorectal cell line, and all of these gave similar results to the one I'm showing on the screen. Now, let's look into these phos in more detail. To optimize cryopreservation further, we needed to understand the differences in the compositions of each of the phos mixtures we tried, and what were the key differences in the ones that had a very good cryoprotective ability and the ones which had a very poor cryoprotective ability. This slide shows the chemical composition of each of the phos molecules from the last slide. We can see that phos mixture 1 and 2 contain gluconate. In the pictures, this is turquoise. This is a byproduct of the manufacturing process and is not very cellular friendly. So it's unsurprising that when we removed it from the mix, it improved the cellular outcome. As we move from phos mixture 3 through to phos mixture 8, we are essentially removing the smaller molecules, the independent fructose and glucose molecules, and enriching the trains of 3 to 5 base sugars in the mixtures. As we move to phos mixture 8 on the bottom right of the screen, we see that we are heavily enriched in DP4. This is the brown segment. That is sugars of 4 molecule bases, with substantial amounts of DP3 in green and DP5 in dark blue. Almost all of the smaller molecules have been removed, so it's likely that protection is offered by these longer chain molecules in our cellular systems. And just summarising from the previous slide, we can see the worst performing phoses contain gluconate. The intermediate doesn't contain gluconate, but it does contain a lot of individual glucose molecules. And the best performing, the one on the right, removes most of the small molecules, leaving a mixture that's primarily composed of phos containing 3, 4 or 5 base sugars. So, of course, there is always much more to say, but to keep in time, I'll start wrapping up here and going through some of the conclusions. FOSS is an interesting option as a future cryoprotectant. It has several key advantages. It needs only cheap starting materials, so it can be easily synthesized. It's not cytotoxic, so it can be added at a higher temperature, and longer cryopreservation times shouldn't result in severe time-dependent toxicity as we sometimes see with DMSO. This also removes the need for a washing step post-thaw, so it makes processes simpler. Of course, these are still early days, but we believe that these results are very promising and encouraging. There are, of course, many steps before these can be used more widely as a cryoprotectant. We need to optimize the concentration we use. In these experiments, we chose 10%, as in some initial screens, 10% worked very well, and 10% is also typical of cryoprotectant concentrations used with other solutions. However, perhaps 5%, 20%, or something in between works a little bit better. What carrier solution should we use? Should we use water, saline, culture medium, or something else? And what will occur if we try using FOSS in combination with other cryoprotectants as part of a wider cryoprotectant cocktail? We'd like to test on other mammalian cell types, particularly primary cells, and also we can determine what further improvements we would need to make to the composition. To summarise, we have found that fructose-based oligosaccharides can act as an effective DMSO-free cryoprotectant for at least some mammalian cell lines, and by optimising the manufacture and biological applications, it could be improved further still. Before wrapping up, I'd like to thank all our collaborators who worked on the project, and also to the EU for funding the Premium project as part of the Horizon 2020 programmes. Finally, thank you all for listening and for watching this presentation.